I just want to say that what I've done is bring one aspect of analysis that we do, and I'm drilling down very deep to sort of give you an idea of that. And in my talk, I'll mention it, but I won't give all the details. So those of you who attended now will have the benefit of knowing how that information is gathered.
Uh,
So what I want to do,
Denise, maybe you can make a presentation mode.
yes, I
Yeah.
should do that.
Yeah.
Thank you.
Cool. Yeah.
Okay. So, let me just move this. Okay. There we go. Let's see if it'll do it. All right. So, so what I want to do is very quickly remind you of traditional dynamical systems, which I know you all know, but I want to use some terminology and I want to be sure that we all know what that terminology is. Okay. So for example, and you could do this problem when you were an undergraduate, probably, what's an example of how you model population growth? Of course, I'm going to do people, but we could do cells, we could do bacteria, we could do rabbits. So what will the population of the US be in 2030, given it's, let's say, 270 million in 1999? I just made these numbers up, so they're probably not even accurate. So we define a population, T, and then we assume that the population will grow proportionally. Of course, you know, that makes sense, right? You have to be proportional to how many people are there because that's how we reproduce. So if I draw a picture for it, and by the way, Mike Savageau was very big in that the most important thing you can do as a modeler is draw a really good picture before you write equations. And so we have a population, and we're going to say it's growing proportional to itself at a rate K. And so so what we do then is we write what that proportionality looks like. A rate, of course, you know, is a derivative, and there's our proportion. So the rate of change is proportional to how much is there, and we're going to call K the growth rate. And that's a parameter. And so parameters for me are everything. And as a person who really cares about making predictions for the biology and the clinical setting, all the answers, I think, lie in the parameters. So what I'm going to talk to you about today is ways to analyze those parameters. So of course, we all know the solution to the simple system looks like the exponential function, where you have some initial population and it's going to grow exponentially at that rate. And as I said, you can do this with any population. And of course, here's the solution for different initial conditions, which of course doesn't make any sense because we know the population is not blowing up. So for those of you who are new graduate students, you know, what are we missing from our model of population growth? Anybody want to say?
Please, yeah, please unmute. Anyone can answer.
Yeah, you were going to talk with me. So anybody?
Yeah, please go ahead. Yeah.
Capacity of the total population.
Oh yeah, capacity. A very easy way to say that is even just death, right? Because people are dying. I don't even have people dying in this population, right? Then capacity is more complexity, all kinds of things like that. So, and in fact, I will tell you guys that there was a guy named Malthus in England in the 1700s who did a model, right? And he said that this was the model of population growth, and food was growing arithmetically, but the population was growing exponentially, so we were all going to die because we were going to run out of food. But of course, a very important thing he lost was some sort of a lost term. And that lost term, as was correctly identified, includes some sort of a carrying capacity. The population can't grow past that. And also a loss because, of course, in this representation of it, logistic, they're pushing against each other, and you know, whether it's bacteria in a dish or the population in a complicated city that's very populated. But now we pick up a new steady state. Before we had P equals 0, which was unstable, but now we actually pick up a stable steady state, P equals C, and you can see that populations above and below grow to that. Okay, so if you want to analyze these kind of models, the power is really in the parameters. They're the rates of the processes, things like velocities, reaction rates, kinetic, binding constants, rate constants, death rates, growth rates. They actually represent key processes in the system, especially if you've done mechanistic modeling. You know, if you do phenomenological modeling, kind of represents a sort of a idea. And if you need more information, you're going to have to drill down, which I'll talk about later. You can create knockouts and depletions, you know, using the parameters, perform perturbations to your system. You can estimate kinetics, maybe if something isn't even known experimentally. And you can even suggest missing elements in your system through these parameters. And what I'm going to talk to you about now is performing something called uncertainty and sensitivity analysis. So here's the idea. how is modeling used for infectious disease trends to assist in guiding policy decisions or scientific research or really anything? I'm talking about infectious disease here because that's the example I'm going to give you. But of course, parameter analysis is used for any type of model. So I'm going to give you the very classical example of an SIR model, which is you divide your population mutually exclusive into susceptibles, infected, and recovery. So in this model, the simplest, you're susceptible, you get infected, and then you recover, and you never are susceptible again. You have lifelong immunity in this really simple example. So maybe measles, mumps, chickenpox maybe isn't the best example because you can get later in life shingles, so maybe that one's not so good, but you are protected somewhat from chickenpox. Okay, so here's the simplest equations I can write. So basically, if an S interacts with an I in a mass action type of setting at a rate beta, it's going to be a loss term here and a gain term to my rate of infection. And then these at rate gamma can recover, and they're in this recovery term. So we want to understand this a little bit better, and you'll understand why I picked this example to go over later. But if I can solve this, of course, and you can see if I solve this, well, you know, the susceptibles die off and everybody becomes recovered, and the infecteds, of course, have this kind of behavior. But let's look at that a little deeper. There's this concept of something called a reproductive number. Some people call it sort of like, you know, the conditions for an epidemic to happen or, you know, or not happen. So let's think about what that means. That means that our rate of change of our infecteds, which is given by this equation, is going to be positive. And that's what's going to drive our rate of infected people up, right? And so if I solve that equation, what I can do is bring this onto the other side, divide both sides by the infecteds, and I'm left with a ratio that basically says beta gamma, if it's bigger than 1, it's your force of infection here. And so beta and gamma, beta are two parameters that are driving all the behavior in the system. Now, this is kind of a silly point because, of course, they're the only parameters in this model. But most of the time, that's not the case, that every single parameter in the model ends up in the R naught or being important. They are here because I made a really simple example. But what I wanted you to see is that these parameters are driving all the behavior. And if you know dynamical systems theory, these parameters are actually comprise a transcritical bifurcation, where basically what happens is, you know, as you change the values, you're going to make this term pass through equal to 1 and then less than 1. And when that happens, of course, at equal to 1, the eigenvalues of the system are 0, and less than 1, the eigenvalues of the system will be negative, so things will be stable, right? And they'll be positive if it's greater than 1. So these parameters are part of a transcritical composite bifurcation because either one of them independently will get you there. Okay, so that was the background for what I want to tell you. Because what if you're not using differential equations? Or even more so, what if you're using differential equations that are really hard to solve? Like, I know people invent all these new ways to find R naughts and study systems and find eigenvalues, but frankly, even MATLAB can't do it all the time. So we need another way to do it. And so that's what I'm going to present to you guys now, something we call uncertainty and sensitivity analysis. So I think that these are key. This is a key analysis tool for a complex system. So uncertainty tells you how much variability are in the model outcomes, so like our susceptibles and our infecteds and our recovereds, based on the changes in the parameters. And we use something called Latin hypercube sampling to help us get that. And then there's sensitivity analysis, which tells us which parameters are inducing all the behavior. Now, in the model we just did, both parameters, there was only 2, caused all of the behavior. But in models that are very complex, you know, I mean, some of our models have hundreds of parameters in it. Certainly, you know, not all of them are driving you to different outcomes, okay? So, and we use something called partial rank correlation and EFAST for that, which I'll tell you about in a little bit. But here's the most amazing thing. We can quantify the relationship over time. So this is what I mean. I can tell you that early on in the epidemic, as it's evolving, maybe gamma is really important. But later on in the pandemic, maybe beta plays a bigger role. So at every single time point, the uncertainty and sensitivity analysis can quantify these two measures for us and this partial rank correlation. And I'll explain to you why it's a partial rank correlation in a little bit. I'm just kind of giving you an overview.
May I pause a little bit
Yes,
here?
please.
Yeah, I'm actually honestly confused about two concepts, uncertainty and sensitivity. It sounds very similar to me.
They do.
Yeah.
They do. So I'm giving you an overview, and I promise in the next slides, as I show you the difference, you'll know exactly what the difference is. Okay.
Thanks.
So hang
Thanks.
on. Okay, and I want to tell you that all of this is published in a paper that was published in 2008, and all of the programs to do these analyses are free and available on our website, and they're both written in MATLAB and also in R. Okay? All right, so I'm going to tell you about the LHS procedure now. So suppose I have K parameters in my model. I had 2 before, but now I'm going to have K because it's more interesting when you have more. Okay? And what I'm going to do is wonder if there's a probability distribution for each. Now, most of the time, I don't have any idea about my parameters, so I'll give a normal distribution. But sometimes, a uniform distribution. But sometimes I do have an idea, and I'll give them a normal distribution. But each parameter comes from some sort of a distribution in its, parameter space. And what that does is create something called a k-dimensional hypercube. And these parameters even include the initial conditions in your model, if you want them to, okay? Now you're going to sample from this parameter space n times. N is really important. We use n like 1,000, but there's actually a formula for computing n. It's greater than 4/3 the number of parameters, okay, that you have, but we usually use much larger than that. So what we're going to do is I'm just going to show you the example with 2 parameters. But we have 2 parameters here, and they range. This one ranges from here to here on a number line, and this one ranges from here to here. So the first thing I'm going to do is I'm going to divide up by n the intervals of this range for each that I'm giving you. Okay? And then I'm going to, and you notice that I have a normal distribution. A uniform distribution applied to the shape here. But we could have had a normal distribution, and if we had, we would change the width of some of the slices because every slice has to be equiprobable, if that makes sense. So uniform, it's easy. They're all equiprobable. But if you have a normal distribution, some of them are going to be skinny and tall. Some of them are going to be fat and wide if they're at the ends. Okay? Okay. So then I'm going to start sampling from this parameter. So now I've only done it for 2 parameters, but you do it for all K parameters that you have. And I'm going to say, okay, my first sample, I chose the value A from parameter 1 and the value X from parameter 2. And for my next sample, I'm going to choose parameter B from here. And by the way, you sample without replacement and until you cover the entire parameter space for each parameter. And now what I've done, guys, is I've created a giant matrix with it's K by N. So everybody see it's K by N. K is the number of parameters, N is how many times I've sampled, and it's every single parameter sort of all mixed up, in different combinations with each other. Everybody got that so far? Yeah. So this is the Latin hypercube sampling uncertainty. All right. So now what I do is, wait, let me see if I did. Sorry, I'm going to go forward. I didn't. Okay. So I was limited on time, so I was seeing like how much to give here. All right. So now what you do is you simulate your model. Right. N times. So I have a row. So row 1, I simulate my model with that. Row 2, I simulate my model with that. Row 3. And so this is the uncertainty. And when it's done, I'm going to see all these different simulations. But I guarantee you they're going to be nested. And some of them are going to go up, maybe. Some of them are going to come down. And the idea is you can start to actually see all the behaviors that your model does within the biologically reasonable ranges that you've assigned to it. Right. And if you see something really weird, then you're going to say, oh, wow, I don't know. I should not be in that part of parameter space. So you might go back and adjust your parameter space. But this is your uncertainty analysis. You sample your parameter space and you look at all the outcomes of your system over time after running it. So now that's uncertainty. So now we'll talk about sensitivity. So sensitivity, first of all, oh, and I have to tell you this, you don't have to vary all the parameters. I could have a parameter number 3 that I pick the value of 4.0, and every single time under that thing, it's 4.0, okay? You don't have to vary all the parameters. You can just, so for the ones that vary, you are defining how to look for significant. Now, here's the idea. When you do regular correlations, you're looking for a linear correlation between variables, but we are doing nonlinear modeling.
Right.
And so partial rank correlation allows you to quantify nonlinear relationships, but they have to be monotonic. That's very important. If they're not monotonic, so suppose you're a person who lives in oscillation world and you have lots of oscillations, then you're going to use the EFAST method, not this partial rank correlation. So you can see that in my paper, you're going to use EFAST and it's going to give you the same kind of results as what I'm getting here. But for most of the models that we do, they're not oscillatory. So we're very good with the monotonic, positive, definite, models. Okay. And so the idea is, instead of you're quantifying and ranking the rankings of the runs as opposed to the values, and that's why it's called a rank correlation as opposed to other, and it's a partial for other reasons. So what it's going to do is give us values between -1 and 1, which I personally believe are transcritical bifurcations, and I'll tell you guys that later, is that I believe that this is our link to traditional dynamical systems theory right here. And you can compute these partial rank correlations at every single time point for your model. So it's really hard, and it changes. Of course, your model changes over time. And EFAST, as I mentioned, is for the monotonic ones. Okay, so here's another thing I have to tell you about. For deterministic models like ODEs, there is one type of uncertainty. And it's uncertainty from the parameters, like we talked about, the LHS variation there. So you put some inputs in, there are some uncertainties, and then that creates uncertainty in the output. For stochastic models, there's not only input uncertainties, but there's also stochastic effects within the model. So that is known as aleatory uncertainty. So the first type is called epistemic uncertainty, and the second one is called aleatory. So it's if you have 2 times. So sometimes, that N, you might have to repeat those same simulations multiple times because you have uncertainty in your system with a different seed and see what happens. So this is all explained in the papers and on our website, but I'm just kind of giving you an overview. The other thing that you can do is you can look for sensitivities if you have a multi-scale compartment within the same compartment that you're interested in, or you can look at sensitivities between compartments. So I can hold fixed one compartment and look at the sensitivity in another compartment, or I can look at it in my compartment. So I could say, suppose I'm holding the compartment of blood fixed, but I can see how it's affecting the lung compartment, for example. And so you can do these sensitivities, and it's very, very powerful. So I'm going to give you an example. So this is a result of a sensitivity analysis that I'm going to show you in a few minutes when we go to the other talk, where I'm showing you only those parameters that were significantly ranked. I'm showing them over time. And I want you to notice that out of the 100 parameters in my model, only very few are sensitive, are actually what we would call bifurcation type parameters that are driving behavior. And you can see, importantly, in the first 50 days, different parameters are involved than later when after 50 days, other parameters become more dominant in driving the dynamics.
I see.
So this is the sensitivity analysis here. The uncertainty analysis vary all the parameters over a wide parameter space and look at all your outcomes. That's uncertainty. Sensitivity says which parameters are giving you all the difference that you're looking at when you did that. And it tells you exactly which ones do it. So before I go on, let me stop here and see. Now I can answer questions because I hopefully gave you a big enough picture and we can go into detail. I know we only have a few minutes, but let's see if there's any questions.
Yeah. So yeah, if you have any question, yeah, please ask. Yeah.
Simply here, if some parameter has higher value, which means the parameter can derive the system differently.
That's a really
slowly.
good question.
Okay.
That's a very good question, Jinsu. So let me answer that for you. In regular correlation analysis, that is true.
You're right.
In linear correlation, that is definitely true. Bigger numbers mean more important, but that's not true when you're doing nonlinear partial rank correlation. We actually have to do another test. So right here, we don't know that this one here is the most negatively correlated. We don't know that for sure. We actually have to do a further test to find out comparing between the sensitivities. So there's one more step that you have to do to say that. Most of the time it is true, but you can't guarantee that it's true. You have to do another, I think it's a Z-test of some type that you have to do to compare that. Okay, very good question.
Thanks.
Mhm.
Can I have a question?
Yes,
Yes.
please.
Hello, I'm really glad to attend this class. So I have a question about the Latin hypercube sampling. So you mentioned about the distribution, but I'm quite concerning about. So what if we set a quite huge distribution, then we might overestimate the PRCC value. So how did you set the distribution?
So that's a really, really good question. The hardest thing we do is set ranges on the parameters. Once you do set the ranges and the distribution, you're pretty much good to go. But that's the hardest part. So let's talk about that. Some of our ranges, Young-suk, range from 10 to the negative fourth to 10 to the fourth, 8 orders of magnitude you can do. And so we actually have a way in the system where you say, is this a logarithmic variation? Or is it a linear? I mean, is it a regular, non-logarithmic variation of a parameter? So you can vary some parameters over log scale, and you can vary some parameters over linear scale. Now, your question is, what if you've gone too big? Before you do the PRCC, you will know the answer to that, because you're going to look at the output of your model and you're going to see all the different simulations for their populations. You're going to say, what is going on with that? That is growing beyond anything biologically feasible. So then you're going to go back and you're going to figure out which you could trace back to which parameters and which run that was from. And you're going to say, oh, it's because I set parameter 7 too big. And so then you're going to narrow that parameter and you're going to do it again. Okay. And then when you're pretty happy with your output, then you move to the PRCC after that. So nothing weird ever happens at that point. Okay. I hope that is clear.
Thank you so much.
Yes.
I really appreciate.
Oh,
Sure.
By the way, Young-suk is a student of the UNO.
Oh, hi. Tell Uno I said hello. She's a good friend of mine.
Yeah.
Really, she's on another meeting, so she's not.
Yeah, no problem. Please tell her I said hello. I know her quite
Okay.
well. Yeah.
Thank you so much.
And Aurelio, hand. Yeah, please.
Yes, Aurelio.
Hi, yeah, thank you so much. Actually, I'm using your method and your code since 2016, I guess.
Okay.
And I still have several questions regarding the PRCC. First is on the parameters, they contain units. So for example, one parameter has a unit of whatever, and the other parameter has a different unit. Then how do you remove this dimension when you look at the PRCC values? And my second question is...
Okay, wait, let me answer that one first, because I'll forget. And so that's a good question. Believe it or not, your parameters are part of the system that you created, probably ODEs maybe, okay? So they're part of the ODEs, so the stoichiometry of your model should be set up beautifully. So the units don't matter when you do the Latin hypercube sampling, because that's already accounted for in the simulations. It's going to use it in the very correct way that you intended it. So it always ties back to the original model. So you can mix inches with miles. It's no problem. Okay.
Yeah. Thank you. And the other question is, normally when you're looking at the sensitivity analysis, it's the effect of the individual parameter to your output. But most of the times parameters are dependent among each other. So is there another layer on that to move forward on this sensitivity analysis?
So that's a good point. If you have a parameter that is dependent on another, so K equals 2 times, P or something, I would not put both K and P in there. I would only vary P or K. I wouldn't vary both. I think that will really cause you some trouble because it is a function of the other. So I would just vary P in that case,
right.
If that makes sense.
Yeah. Thank you. Thank you so much.
We don't vary out of the 100 parameters. When we do a sensitivity analysis, we don't vary all of them all the time.
Right.
Now, Jay, I know I only have a few minutes left. So what should I do? Should I end or should I say one more thing?
Maybe, yeah, we can enjoy 2 more minutes your tutorial.
2 more minutes. Okay.
Yeah.
All right. So what I want to tell you guys, after I just spent all this time talking to you about LHS, this Latin hypercube sampling, somebody asked us one day, that's not the only sampling scheme out there. What if you use a different sampling scheme that might be better? And we were like, oh no. So of course there's just random. And then there's something called Sobol sequences, which is a deterministic sampling.
Right.
Okay. So I want to just give you 1 minute on this. And we have a paper on this so you can read the paper. But here's what random would look like. Here's Latin hypercube sampling and here's Sobol. And you can see that computational time, if you're comparing it, pretty similar, a little bit better, for the Sobol and random, a little bit longer for the LHS. But we did a comparison of all the advantages and disadvantages of these two sequences, and it turns out that and we did it just for this simple model. You can see that actually you don't learn much more from this really complex approach of changing it to deterministic sampling. So and even the errors are about the same, a little bit better for the errors with the Sobol than most. So what we concluded was that when you would want to use Sobol is, let's say you're not sure how many samples. Like, remember I talked to you about N, we have to do N samples. What if you wanted to do 25 more? In LHS, you'd have to go back and run the whole thing again, adding 25 on. But for Sobol, you could start where you left off and just add 25 more samples to the previous study. So that's really, in the end, what's the best. For calibration and debugging, Sobol is probably a little bit better. So you all can see the paper on that. So I just want to tell you that we believe that traditional dynamical systems theory that's applied to differential equations and is complex. You can use uncertainty and sensitivity analysis, and we believe you'll get a lot of the same predictions this way. And if you use EFAST, we believe you're finding the hop bifurcations, for example. Okay. So, but the question remains: is there a mathematical map between between traditional dynamical systems bifurcation theory and sensitivity analysis that I've shown you today? I believe that there is, because in the cases where I could check, they always showed up as the same for both. But of course, that's not a proof. So there's an open question for any of you really mathy guys out there. And then, as I mentioned to you, if you go to my website, we have a section there on this, and all the papers are there, and all the programs are downloadable.
Wow.
So we're done.
Cool.
Thank you.
Wow, it was really wonderful tutorial. I think so far you got the speaker who got the most questions from the two. That means really good sign. So thanks so much. I think it's a really good example for the future speaker, I think. So I think maybe I can give you 1 minute for your break.
Yeah, I'll just drink some tea and then I'll be.
Yeah. So wow. So yeah, I think everyone, I think we should handle her paper as our journal club, Aurelio. I think you look like an expert, so maybe next.
Sorry, Aurelio, I didn't mean to get you in trouble there.
Cool. So maybe you want to change your slide, right?
Yes, I am, as we speak. And do you want me to share yet, Jay?
Yeah, please share it.
Okay, yes.
And then,
all right. And I can go into presentation mode.
Yes.
Okay, there we go.
Okay, then. Okay, so then, yeah, I know it's weird, but let me introduce her one more time. So, hello, everyone. Welcome to our BIMAC Colloquium series. I have the great pleasure of introducing Professor Denise Kirschner as today's BIMAC Colloquium speaker. Actually, she was one of my teacher when I was at Michigan as a graduate student. In a series of landmark study, Dr. Kirschner has investigated host pathogen interaction in infection disease, leading to immune responses by using multi-scale modeling approaches. And Dr. Kirschner is currently a professor of microbiology and immunology at the University of Michigan, Ann Arbor. And she is also co-editor-in-chief of the Journal of Theoretical Biology. And she was a former president of Society for Mathematical Biology. And actually at that time, I heard that she helped the Philippine Mathematical Biology Society a lot. And then her most recent research one, and the topic of her talk today is system biology approach using multi-scale modeling to understand the immune response during the tuberculosis, TB infection and TB infection and treatment. So please join me in welcoming Dr. Kirschner.
Thank you so much, Jay.
Wow.
This is really an honor to be here today. I'm really grateful for the opportunity to speak to you all, and I'm always available, during the talk for questions. Afterwards, feel free to email me. I'm really excited to be a part of this community. So I'm going to talk about a lot of different things. Hopefully some of them will resonate with each of you, and you'll be, you'll get something out of all the different areas I'm going to talk about. But I want to start by getting on some of the terminology, so we're on the same page. So multi-scale modeling to me refers to a style of modeling in which multiple models at different scales are used to simultaneously describe a system. And the model usually focuses on different scales of resolution. So we'll talk about that in a minute. And I believe the need for multi-scale modeling has arisen because complex biological problems can't be accurately captured at a single biological scale, right? Even though experimentalists all the time do stuff in a petri dish, that doesn't mean that that's the best scale to do it at, right? And so same thing with modeling. I think we have to get beyond just modeling at a single scale. And so the idea is that if you're interested in something about, something about the whole body, there's the organ scale, there's the cellular scale, there's the molecular scale, there's the gene scale, all put inside the individual. And of course, that's even at the population. So there's many, many different levels of, and a lot of people do modeling at one of these scales. What we're suggesting is that connecting these scales is extremely important because people talk about epigenetics. Changing something at the gene scale is going to affect something at the individual scale, right? And some similarly in other examples there. So what are some of the issues that you have to think about? I think the first thing you have to think about is the biological scale and what modeling type might be the best to represent it. So for example, at the genetic or molecular scale, the time scales are something very quick, 10 seconds or something, maybe even shorter. The length scale, very tiny. Intracellular scale is maybe a little bit longer than the gene molecular scale. Again, length scale is small. The tissue scale is things happening at a much slower, more on the minutes and hours, and then of course a bigger scale, cells are a little bit larger, and then at the organ scale, things are happening up to years, and population as well, and different scales there. And of course, there's a different type of mathematics that might well represent this, but of course we're not limited to this. So for example, we think about maybe just deterministic and continuous for these kinds of scales. Tissues, it might be better to track things moving around in them, and so discrete stochastic might be. The whole organism, like I just showed you in the SIR model, and the population, these could be deterministic or a mixed type of a model. And then so statistical models, ODEs, maybe something called individual-based modeling or agent-based modeling. This might be hybrid, types models where you mix the two together. So these are all important considerations when you're trying to build a multi-scale model. So I want to tell you about our approach. What we do is we build models typically start at the mesoscale. Meso means middle. So we're really, I don't know why that little star is there. I have no idea. And so the mesoscale is kind of like the middle scale, and it's usually the scale that you're most interested in. And then, but you're constantly iterating and updating that model with data. You have to choose the appropriate model. You have to calibrate it and validate it. You explore the emergent behavior in that model. But then you might say it's really important to spread out, north and south, build larger scale models or smaller scale models, or even east and west, other models at the same scale. And then you have to think about linking them, which is not a trivial thing. And that's actually quite model dependent. And then on top of it, you have feed forward information and feedback information. So as we talked about, the gene scale might influence the population scale, and also the population might influence the genes, right? So, and then of course we want to perform analysis on these, which those of you who came early got to hear about that. And then I want to talk to you just for a second about something called Tuneable Resolution. And then, and we've done work in parameter identifiability and our ODEs and PDE models, but I don't have time to talk about those now. So you can just see the website for papers on that. All right, so I'm going to use as my example tuberculosis. It's the #1 cause of disease in the world today, even despite COVID. So in the past 2 years, 2 million people have died each year from COVID, but in the past 50 years, 2 million people have died a year from tuberculosis. So 1/3 of the world's population is infected with TB, which is pretty impressive. So upon a chest x-ray, you can see that there's a nodule in the lung that's called a granuloma, and these are what the bacteria look like. And as a mathematician, what got me interested in studying TB was that some percent of people get exposed and clear. About 5 to 10% get primary active disease, where if they're not treated, they'll die. But most are 90% control disease, which is clinically latent, but not, and it's not infectious, but that doesn't mean it's a dormant disease. And so over the lifetime, it can reactivate due to HIV, immunosuppressive drugs, even COVID, and you end up with active disease again, which you have to control through drugs, or you'll die. Okay, so what am I interested in? I'm interested in the lungs because bacteria are breathed into the lungs, and then local cells called dendritic cells and macrophages take up that bacteria. But think about this bacteria has been around since the time of Egyptian mummies. I'll explain this to you. So you all have heard of leprosy, biblical disease, right? It's been around since BC times. And it's called Mycobacterium leprae. This is Mycobacterium tuberculosis. So they are the same species, but a different, same genus, different species. And also we find Egyptian mummies with tubercles in their lungs. So TB is a very ancient disease. So imagine this bacteria has evolved dramatically over these thousands and thousands of years. So it grows happily inside the big eater cells, macrophages, that usually eat it. And actually once it reaches capacity, it'll burst it open. New macrophages will take it up, and this process will continue. Eventually, a dendritic cell will take some of that bacteria to a lymph node, sorry, it's late, prime these prime T cells to go back to the lung and then activate those macrophages. So if the T cells are around, those same macrophages which couldn't do anything become activated and then can kill their intracellular bug, or those macrophages themselves can be killed and the bugs inside them along with it. But the interesting thing is the signature in the lung, something called a granuloma, and it's a conglomeration of all of these different cells, dead cells, live cells, and it has a very characteristic function. And I will tell you that more than one form, because usually you breathe in more than one bacteria. So this is just an example from a monkey, non-human primate, of 4 different types of granulomas that form. So these granulomas form in the lungs, and they have lots of different looks to them. And so modeling, I think, can maybe help distinguish all of this. So we are very lucky to have a colony of 300 monkeys paid for by Bill and Melinda Gates Foundation that are housed at the University of Pittsburgh, where we we infect them with a very low dose of bacteria, and we get about a 50-50 split in these monkeys. Now, remember I told you in humans it was 90-10. The monkeys are more of a 50-50 split of those that get clinically latent versus clinically active disease. So that gives us a larger sample size to work with and get data from. But here's the interesting thing. If I look at one monkey number 19608, he had 5 granulomas. And there's a dye that you can give to the monkeys that their granulomas will take them, and you can look at the granulomas through an X-ray, okay? And a PET CT scan is what it is. And you can see that I've labeled them each by a different color. Well, it starts out, some are very bright, some are very not as bright, some are smaller, some are larger. But look what happens over time. And if I trace the trajectories of each of those granulomas, each one is an island. It does its own thing. And it doesn't even care what the other granulomas are doing. So this allows us to really study TB at a granuloma scale. Because if we can understand what's going on in each of these granulomas, then we can probably have a better idea collectively of what's happening. So the first question is then, if we're going to try to model these granulomas, what are we going to do? Well, 20 years ago, we tried ODEs, then we tried PDEs, we tried something called metapopulation modeling, and then about 15 years ago, we settled on using agent-based modeling and individual-based modeling for this. And we wrote a very interesting paper in 2005 where we compared what you learn from using these different modeling approaches and what are the pros and cons of using each. So that might be of interest if you're not really sure what the best modeling approach is for your work. So an agent-based model, I believe, has 4 things. The environment that you're performing the model on. And so here I'm doing it by 2 millimeter of 2 millimeter lung tissue. We actually changed the millimeters here. It can be up to 4 or 6 or 8, but it's a chunk of lung tissue. The agents, which are really the variables in our model. So the immune cells, the macrophages. And then, of course, there's some continuous agents, things like cytokines, chemokines. But these are discrete. We're counting them individually. There are different types of cells. There's also things that are secreted. They diffuse on the grid. There's also vascular sources in the lung tissue where things can come in or go out. And then there's the rules about how all of these different things interact and the time steps at which they operate on. So agent-based models are the 4 things in red: the environment, the agents, the rules, and the time steps.
I see.
Okay. All right. So I'm going to talk to you today about the agent-based model we created called GranSim for Granuloma Simulator. And it's a cellular tissue scale model, which means that we put in the rules, all the biological rules for cellular scale interactions, and the tissue scale is where the answers emerge. So this is emergent behavior in this model because this is a very complex biological problem. And so we give all of these rules that are checked by biology, cell movement on the grid, interaction between T cells and the macrophages, secretion of cytokines, all of these things are in our model. And remember, we talked about building them. So we have the molecular and the cellular scale that leads to a granuloma scale outcome. But of course, that's inside of lungs, which are interacting with blood and lymph nodes, as I showed you earlier. So it's a very complex system. But I started at the meso scale. I cared about what was happening in the granuloma. And for me, that was the tissue scale level. So I built a cellular scale model that gave me the tissue level outcomes. Okay. So this is an example of a granuloma taken from a monkey, and it's been sliced and stained for 3 different types of cells. It's been stained for macrophages, for all kinds of T cells, and for neutrophils. And you can see that they form this very characteristic structure. See, when we started modeling TB with ODEs 20 years ago, we didn't have this kind of data. So we thought that they were all just mixed up in there. But you can see there's a very important organization. And we think that the cellular spatial structure is incredibly important to the fact that these granulomas do a darn good job of controlling the bacteria in them most of the time. If 2 billion people in the world have TB right now, but only a few have active disease, it's these granulomas that are making that happen. Okay? And in the center of all granulomas is caseum, or something called caseous necrosis, which means dead tissue. very similar to a tumor. A tumor forms and the center becomes necrotic because there's no oxygen and there's no blood supplying it, right? And the same thing happens in granulomas. So a lot of what I'm telling you today can also be explored for tumors as well. And so here is our computer simulation of granulomas. And so we did not tell it to draw a granuloma. We gave it the rules. And yet this is the emergent behavior that we saw. And it's the emergent behavior every time we run GranSim. So it's obviously a very stable attractor for those of you that are dynamical systems people. This granuloma, both biologically as well as computationally, is a stable attractor. And you can see this ring of lymphocytes. Of course, the T cells are small pink ones because they're smaller than the macrophages, which are green and blue. And there's a lot of dead tissue in the middle, bacteria trapped in different cells here. And that's what we see happening. But of course, we don't need the picture. What we really want is to track this developing over time. And so we can do that. We can do a time-lapse video of dropping in a single bacteria. And these, the green are the macrophages that are just in your lung normally. The little pink squares are your vascular sources where T cells can be recruited. And you can see that this granuloma, so this is a 200-day simulation that I'm going to show you in a matter of seconds for this granuloma to form in our lung tissue.
Wow.
Okay.
I see.
All right.
Wow.
So of course we put our model through many different calibration rules. And so here's an example of lots of 600 granulomas from non-human primates sacrificed at different times. And you can see the range and the median on their granulomas. And so of course our granulomas match within that same range. But as modelers know, we can go beyond and predict more of what's going to happen over time, right? But I also want to tell you that everything I've showed you is in 2D. A lot of people say, well, Denise, granulomas are 3D structures. So we have built a 3D version of GranSim. Regular GranSim takes about a half an hour to run. 3D version takes about 7 days. So you can imagine that we don't run this model very often unless we really need to. And you can see I'm sliding a level set through this space here to show you what one of the molecules looks like over time. So that's just an example of how, and this is, we use a beautiful simulation program here called Data Tank, which is free. But we don't run this model most of the time because again, it's very computationally time expensive. I will tell you 2 things before I move on. One is that we've built a way to compare 2D versus 3D. When is it okay to use a 2D model of a 3D structure? And how do you take 3D data and use it in a 2D model? So we have 2 papers on that topic and you can find them on our website if that's something that's interesting to you. Okay, so now the next question is, and this is important for many modelers, is how do we calibrate to lots of different types of data? Okay, so the first thing you should ask yourself is, what are you calibrating to? If you're calibrating to a single trend line for your outcomes, then you can just use typical, probably least squares kinds of things, objective optimizing tools. But if you have a range of data for lots of different time points and lots of different data you're trying to fit, you have to ask yourself, do you feel comfortable assigning a distribution to that observed data? If you are, then you can use things like Bayesian calibration or samplets, important resamplings kinds of approaches. But if you don't, we've created a program called CaliPro, which can help you calibrate your model. And so very briefly, one slide here is that, you have user input, you sample, here I sampled using Latin hyperacute sampling, but you sample your space, for your different parameters that you have. And then you run your model and you decide pass-fail criteria using ADS or HDR, and that allows you to iterate this process until you really hone in on the parameter space that you want to use. So somebody asked me that question in the first talk about parameters. CaliPro would be a good way to help you find the actual parameter range that you think is most relevant for the biological system that you're studying. Okay. And this paper has appeared in Cell and Molecular Bioengineering. And again, it's all on our website. So, you guys saw this plot earlier, those of you who watched. So I used my fabulous uncertainty and sensitivity approach to look at GranSim and say, which out of all these interactions in your model, which parameters are driving all the behavior? And what we noticed, surprisingly, is what parameters that were involved in generating the granuloma are not the same parameters needed to maintain that granuloma over time. In fact, this one started out to be positively correlated and ended up to be negatively correlated, right? Very important with the outcome of a bacterial load in the model. So my outcome variable here is bacterial load. The other thing I noticed about these 8 parameters that showed up is that they fell into 5 categories. Some involving a molecule called tumor necrosis factor, or TNF, the rates that you recruit your T cells and how T cells move on the grid, your macrophage activation rates, your intracellular bacteria growth rates, and the dynamics of how you build that dead tissue in the center of the granuloma, all figured out how much, whether that granuloma was going to control infection or that granuloma was going to break open and do a really bad job of controlling infection.
Mm-hmm.
So now here is where multi-scale modeling comes in. So far I've talked to you about things that are happening at the cellular scale and with a few molecules diffusing, and then at the tissue scale. But I've just been told that many of the dynamics depend on what's going on with this molecule. So I can't tell them TNF is important, because guess what? They already know that, because there are situations where, okay, so I deleted a slide. Sorry. There are situations where you take a mouse and you can deplete TNF and they will get active disease. You can even give humans anti-TNF drugs to treat rheumatoid arthritis or Crohn's disease, and they will get active TB because they had latent TB that was reactivated by mistake. So this is stuff that they already knew. And look in GranSim, if I create a virtual knockout with no tumor necrosis factor, this same granuloma falls apart and begins to spread bacteria and grow really bad. So of course TNF is important, but I want to give them mechanism. So to give them mechanism, I want to be able to drill down now and build a very detailed molecular model and connect it to my cellular scale model. Okay, so that means I'm going to build a model of macrophages that have TNF receptors on the surface. There's 2 types that internalize, degrade, synthesize, reproduce, all the things that happen here. It's a receptor ligand, basically scale model, and that is going to run inside every single macrophage in my agent-based model. Okay, that's a lot, right? It already takes 30 minutes to run GranSim, and now you're going to run, 15 equations inside of every one of the macrophages in this model that are processing what's going on with this molecule TNF. And so, there's 9. Sorry, there's 9 ODEs that create this TNF submodel that describe that. I won't go into that today, but it's very complex. But let me show you what we learned. We learned that one parameter, the TNF molecule receptor 1 internalization rate, is a bifurcation parameter. Because if it's very, very small and not doing anything, I get clearance of all the bacteria, but look at all those immune cells in the lung. Very bad. Inflammation in the lung is something you never want. That'll lead to pneumonia. If it's in the middle range, you end up with sort of granulomas that look like what we're used to. And if it's in the high range, you get uncontrolled growth and the bacteria just take over. So this is a parameter that can't be studied in the wet lab. But I've just told them that they have a handle for TB. If they can create a drug that targets this TNF receptor internalization rate, they would be able to make all these granulomas, good granulomas, as an example. So does everybody sort of get how I did that? Any questions? I'm happy to stop here and take a question because this is complicated stuff, I know.
I think so.
Is that okay?
If you have any question, you can unmute, ask a question.
If not, we'll just keep going.
Yeah, but I think it looks
okay.
Okay, yeah, please, yeah.
Okay, So now I've told you guys that, oh, sorry, I've created this really complex model that's running inside of all my macrophages. But, you know, if I'm not asking questions specific to TNF and what it's doing, do I really need all that turned on running all the time? The answer is no. But can I go back to where I was before I built this submodel when I was dumb? Because I didn't know all these details about TNF. Now I got really smart because I'm like, oh, look at the way TNF behaves and all this stuff. So when I go back, I can't just shut it off. I have to go back smart. And so I have to shut all those dynamics off using what I learned from building this very complex model. And so I do that now. And so I call that Tuneable Resolution. So this is a paper that that we published about, I don't know, 8 years ago. And I'll tell you the idea here. It's a way to deal with this increasing model complexity and numerical issues, because again, I told you running those submodels took a long time. So it allows us to fine grain or coarse grain a model at will. So if we have a submodel, let's say we're running it and we don't need it on, but if something happens, we can actually have the program say, oh, time to turn it on, and it can actually turn on and run in the model and shut it off if we want to. It can adapt to the degree of detail that you want to ask, and it can assist in model analysis, computational speed, and debug. But the important thing I want to tell you is that Tuneable Resolution is not model reduction. Model reduction is a mathematical concept that you can create a smaller model that is mathematically isomorphic to the bigger model. Tuneable Resolution says we want to create a smaller model that's biologically isomorphic to the larger model. Does everybody see the difference there? So the math might be very similar, might be very different, but what we're trying to do is make a very simple version that captures the key piece of biology that I learned from that model, and so I can include it. So, you guys will have to see our paper on that. You can go to our website. We kind of walk you through a method about how to do this with your modeling. If you have a very complex multi-scale model that you want to, you know, just really focus on one of the scales and kind of tune out the other scales, if you will. All right, so the last piece I'm going to tell you in the time that we have left is about treatment. And one of the reasons I'm going to tell you about treatment is because there's a lot of things that are interesting in mathematical approaches here, but also I think it's very amenable to those of you that study drugs in other systems, in tumor systems, or in other diseases. So I think you'll find this interesting. So why is TB so hard to treat? Well, the first thing is you have a bacteria that grows very slowly. Most bacteria double every 20 minutes, like E. coli, but TB doubles every 24 to 48 hours. So it takes 2 days for a bacteria to divide into two, Mycobacteria. All right, that's incredible. That alone helps this bacteria survive the way it does. The second thing is we've got these granulomas, which are a physiological barrier. So if you give antibiotics, here's one drug, it doesn't get into this necrotic core. This drug does get in, but it's not as much as that it's in the outside. So you have this heterogeneous environment where you want these drugs to penetrate. And it's patient-unfriendly treatment. Because of number 1 and number 2, we give people 9 months of multiple antibiotics, right? How many people don't even finish their 10-day course of antibiotics that the doctor gives you, right? You don't even finish that sometimes. So are you going to take 9 months, 4 different antibiotics every day, lots of side effects, right? So those are all the issues with treatment for TB.
I see.
So the first thing we want to do is we want to build what's known as a pharmacokinetic and pharmacodynamic model and put that in GranSim. So the idea is we have a transit compartment. Some drugs have 1 or 2 compartments depending on the type of drug. So that means you swallow it, it goes into your stomach, then maybe it goes into your blood, then it transfers to another area, and then it can come through the vascular sources in our model and get onto the granuloma, okay? And once it gets there, we assume that the antibiotics kill is proportional to the drug concentration that's there in some kind of a Hill constant way. Okay. All right. So if I simulate GranSim, I've showed you that there's kind of 2 types of granulomas. Granulomas that are doing bad and growing big and their bacterial load is very high, and some that are very stable and controlling. That's what we want. And so here's an example. Here's one that looks you know, good, and this granuloma probably be with this person their whole life. This one though, yuck, right? It's getting big. It almost looks like it's pinching off into 2 granulomas and really spreading. So it turns out that treating people that have these good granulomas is very easy. The drugs work quite well. But even if they have just one bad granuloma, it's very hard to treat them. So what we really want to do is treat these bad granulomas. And that's what I'm going to talk about now. So let me show you what it looks like when we run our simulation. If we dose one drug, 7 doses in a week. So every day we gave a drug. And here it is. It's going up and down, right? Because there's a half-life. Now, what I'm showing you here is the C50s for the, which means the effect that you kill half of the bacteria, the concentration where you kill half of the bacteria, for the intracellular bugs, the bugs that are trapped around, and then the bugs that are trapped inside that dead region. This drug hardly ever gets those guys, right? And so you can see that it's very slowly affecting the concentration. So after 1 week, we still have not even made much of a dent on the bacteria during that time. You can see there's still like hundreds of bacteria in this granuloma. So here is a simulation. So here's a granuloma, and this is another thing that's really helpful with modeling. I can treat that same exact granuloma. If I use the same seed in my simulation, I can treat it with 2 different drugs and see what happens. So if you look, I'm going to treat it with one drug called RIF and another one called INH. And so the INH is already gone after I gave it. But the RIF is staying around a little bit longer until I give the second dose, and then it will accumulate just a little bit more. Here comes the second dose of drug for both of them. It'll be here in a second. Where's the second dose? See, this one is really diffusing, and now here comes the second dose. But there's something that you should notice about this. You know how it's hardly, the drug is barely getting into the center of that granuloma? No wonder it takes 9 months to treat, because you have to continue to bombard these granulomas with drugs to get the drug to kill everything that might be trapped inside of it. All the drug is sort of wasting outside. And that's something that until I did these simulations, people that study TB didn't even realize. So we can do things like treat with multiple drugs. So here's treating with 4 different TB drugs. Oh, I'm going to show you the bad granulomas. So here's the high CFU granuloma, the high bacterial load granulomas. I can treat with 4 drugs individually. They don't do much at all. But if I combine them together, you can see after 180 days, after 6 months, you're starting to sterilize now. The bacteria are being cleared. The good granulomas do really well, and they sterilize much earlier. But these bad ones, but you see, they don't have a way to predict how these drug combinations are going to work in TB. And there are dozens of drugs for TB. And because there's so many drugs, like let's say 10 drugs, you can have an early treatment segment and a late treatment segment. You can give up to 4 drugs per segment. You have 5 possible doses to pick from, let's say. You can give it once a week, twice a week, 3 times a week, every day of the week. If you look at that, that is 10 to the 17th option. Even with modeling, we can't simulate 10 to the 17th, right? Not possible. So this becomes an optimization problem for us that we're trying to do. So we've decided, well, we thought we would use genetic algorithms. It turns out they weren't so good. So we adopted something called surrogate assisted optimization to help us find what's the optimal drug regimens given the amount of drugs that we have. So for example, you have an objective function. Suppose you want to minimize how quickly you clear the bacteria and how much dose you give, right? Those are 2 obvious things. Speed to clear the bugs and the dose. Well, when you do that, we're going to sort of look at it on a surface, like a surface map. Like you can imagine, we're looking for the minimum amount of drug to give to attain this objective. So it's like a topographic map. We really want to hit that objective there. But of course, it's not unique. So there's going to be multiple local minima instead of just a single maximum global. So here is an example using the genetic algorithm. You can see I hit the global max immediately. I did a problem that I already knew the answer to, so I would to test these. And you can see here, it took a while using this surrogate assisted optimization. Did okay, but not great. We improved it. We did much better. But I want to show you that the genetic algorithm took 4,000 simulations of GranSim to get that answer. And this was just with one drug. And so if I use this surrogate assisted optimization with some other methods, I can get very close. And actually I can get multiple local minima that I'd be happy with getting here. And that only took 21 simulations. Okay. All right. So so we have a pipeline now with GranSim where we run GranSim, we find the PK and PD of whatever antibiotics we're interested in. We run them through GranSim with different objective functions. When we build these surrogate models that help us predict, then we do some virtual clinical trials, and then we actually test them in the monkeys. So this is kind of where we're at now. So I just want to finish by telling you one more thing. So we talked early about the fact that there are multiple granulomas. So this is a PET CT scan of a monkey, and here's his lymph nodes, and here are 23 different granulomas that formed in this monkey. Now, let me tell you something interesting. Every granuloma formed from a single bacteria. So they basically blew into a monkey's lungs 23 bacteria, and each one created its own granuloma. Isn't that amazing? And so what that says is that every granuloma arises from a single bacteria. And we can see where they are. They all sort of ended up in this lower left lobe of the lung. But here's something interesting. Looking at them this way, here's at 4 weeks post-infection, you can see one granuloma, and later we can see it split into two. So we have to be able to model. Up till now, I've been talking to you guys about a single granuloma, but what about dealing with multiple granulomas and the fact that there's draining lymph nodes that remember I told you the dendritic cells take the bacteria to the lymph nodes. And so you can see that there's lots of different types of bacteria in the lymph nodes because all these granulomas sent dendritic cells to the lymph nodes to generate T cells to help fight the infection. So basically what I'm telling you is that we spent a lot of time talking about the granuloma scale, but we have to worry about scaling to the host, and then we want to scale to the population. So we can do things like perform virtual clinical trials to make some predictions about things. So to do that, I need not only lungs, but I need lymph nodes, and I need the blood where all the cells travel through. And for TB, pulmonary TB, this is a really good picture of what organs are involved. If it's pulmonary TB, you don't find TB in the brain or in the liver or in the kidneys and other places. Certainly bacteria can disseminate in bad infection, but for most of the TB in the world, pulmonary TB, these are the three organs that we care about. So what we've done now is we've built a model. We took some monkey lungs and we digitized every XYZ coordinate of that lung, and we have blood vessels, we have lymph nodes, and now we can instill granulomas into these lungs so we can have a full picture of what TB looks like. So what you're going to see now is a simulation where we instill some bacteria in this left lung, and you'll see that after the granulomas take about 4 weeks to develop, they're starting to develop now. And the granulomas, the lymph nodes are starting to get hot because cells are traveling there. Then they're traveling back through the blood to the lungs to try to fight infection. In this situation, it didn't do a very good job, and the granulomas are not controlling. And also, it's repeating. Also, you'll see it even spreads to the opposite lung, which can happen. You see these original granulomas, it will actually begin to seed another lung because it's getting into the bloodstream. So now we're able to look at a whole host video and really ask questions about drug treatment, about vaccines, and then we can run many of these hosts to do a virtual clinical trial. So our goal really in the end is to build a virtual human, and it would be amazing to be able to go over all the different scales. We're working on lungs and the lymph nodes in the lungs and the blood, but there's people that are working on kidneys and heart models, and so brain models. It would be amazing to connect all these models up, right, and build a whole virtual human. And then you could ask lots of questions about different sorts of things that you're interested in. So in summary, we've talked that models are increasingly complex, and these multi-scale and multi-compartment models are becoming very popular. We see them in the literature now more and more. We need tools to help solve and analyze them. We talked a little bit today about our analyses. I didn't get to talk to you about how you can efficiently numerically implement and link these scales. We didn't even talk about how you link models. It's sometimes through the parameters, sometimes through other things. We talked a little bit about Tuneable Resolution as a way to help do that. It's very important that you build and validate your models with data. We update our model regularly with new data as it comes in and recalibrate GranSim. And that's where we got the idea for CaliPro, because we had to keep recalibrating the model. We tend to build models from the middle out, the mesoscale out, but they can be built anyway. But usually you pick the layer of interest and then you build out from there as needed. And then you can use models in certainly many areas of biomedicine. And we have an amazing group of collaborators at different universities, many at Michigan, Sriram and biomedical engineering, Jennifer at chemical. We have people at other universities, and we have really generous funding from these different organizations. And here's a picture of us during COVID, all working together. And so thank you so much for your time and attention. And I'm happy to stop sharing and answer some questions from you guys.
Wow, thanks so much. Well, okay, so yeah, it's a question time. So yeah,